nicknamed powerhouses of the cell. The main reason is pretty simple. They produce most of the cell's ATP. A couple of things that are really interesting, and this is one of those interesting things, and that is that the mitochondria contain 37 genes, and this DNA is that of the mitochondria. I guess you can argue it's ours because it's inside of us, but it's not in the nucleus. This is not our nuclear DNA. This is really not what you inherited from both of your parents. As a matter of fact, every one of you only has what is called maternal mitochondrial DNA, only from your mother, which is kind of interesting. The DNA is circular. What's significant about that? Bacterial DNA is circular. That's strange. Bacteria in us. There is a theory, more than a theory, I would say pretty well accepted by a lot of people, that the mitochondria somehow got incorporated into us thousands of years ago. Oxygen was not present all the time on the Earth. At some point, oxygen showed up. Meanwhile, there were cells that were functioning. They were functioning with the glycolytic pathway, a different pathway to make energy. Somehow, and that's not explained how, but somehow the mitochondria got captured or incorporated into some cells. They kind of liked it there because it was the right temperature. Food was provided. Housing, sounds crazy, but in a way that's what happened. And they stayed. And we still have them. Now, how do we know this? Well, there was a person, her name was Margulis is her last name. You can look her up on the internet. She passed away, but she had some of these, at first glance, outrageous theories. She had them early, and she was the one who first came up with this and had a whole set of, let's say, research and other information that led her to this belief. And pretty well accepted by most people. And of course, when you look at this is some indication where these genes come from. Our DNA does not code for them. So the mitochondria also self-replicate. There's no doubt they don't make any mistake. They're not independent in us. Our DNA is regulating the show, but there's at least some portions of the DNA, of the mitochondria's function that's being regulated by the DNA, which is really the mitochondrial DNA. But there is definitely a linking, if you will, between our DNA that is directing the show and the mitochondrial DNA, because there are quite a number of enzymes in here, and there's no way 37 genes could possibly code for all of the enzymes in the mitochondria, code for all the proteins in the membrane, and all these other structures. So it's a lot more complicated than that, but definitely it's kind of interesting to think that they might have got incorporated into us quite a long time ago. The mitochondria has a double membrane. Notice how this outer portion, and then you can see, if I can just follow with the ruler, and then it just goes in and out. I don't know, they did, I guess, a halfway decent job in terms of drawing it. And you see that this membrane has formed shells. These shells are referred to as cristae, that would be plural, or if I said one shelf, I would use the phrase crista. And notice that there are enzymes on or in these shells. These enzymes are referred to as oxidative enzymes. Notice that word, oxidative. So we know that the mitochondria are what are called aerobic. They utilize oxygen. That's why you're breathing, really, is to get oxygen there. If it wasn't for the mitochondria, I don't know what we would look like, but it'd be very different than this. That's what it's all about. Us breathing is to get oxygen to the mitochondria so they can produce ATP, the famous energy currency of the cell. Without the ATP, which is very little we can do. We can't synthesize, we can't transport. That alone is enough to do us in, if you will. We have to have these functions. Just another picture. This time, a little bit of the chemistry is shown. We see glucose, common molecule that is utilized by the mitochondria. It enters through a series of reactions. These reactions are referred to as glycolysis. Then we have the TCA, tricarboxylic acid cycle. I commonly call it the Krebs cycle with a capital K. It's just a number of different things that it's called. And a lot of people know it as the Krebs cycle. This, I'm looking for it, they don't seem to show it, but this is an aerobic process. Not this. This is occurring out in the cytosol and does not require oxygen. As a matter of fact, it's called an anaerobic system. We're going to be looking at this and this again when we get to muscle. Yes?
That's cellular respiration.
Yes. Notice that all they're doing is showing us lots of ATP. In the net production of ATP in glycolysis is 2 for one glucose molecule, and according to this, I've seen 34, I've seen 36, I'll go with 32. 32 ATP. So that's a lot of energy when you compare the two. Which system is producing more? The glycolytic pathway, that's this. By the way, this is 9 reactions, at least 9 reactions. And this, of course, is a whole circular set of reactions with control points. Pretty complicated. Most people, biology students, kind of hate this because at some point, usually they're memorizing the Krebs cycle and glycolytic pathway. Definitely, if you're biochemistry, you've gone into all of this with the control points and everything else. It's just a lot of information there. How do we know about these pathways? We talked about isotopes. We could radioactively label specific carbons, and so we really do know which carbon goes where. I mentioned the fact that they require this. The mitochondria require oxygen. Without it, they die, or they stop functioning, and we die. A long time ago, I think, so one of my students in the other class, I think, looked it up. I think it was in, was it 1982? Or I don't think, because we didn't cover the mitochondria, did we? No? That's what I thought. That's why we're doing it, but it seems familiar to me. Anyway, one of my students in another class this semester, we looked it up together, and I think it was in 1982, there was a poisoning. I don't know what the state it was, but someone actually took a Tylenol package or container, somehow injected some cyanide into it. This happened in several different places. I think there were 7 or 8 people that were killed. And what they did is they put cyanide into the Tylenol pills. The individuals, of course, took the Tylenol and they died, of course, of cyanide poisoning. Well, what does cyanide do? It poisons basically the pathways that are involved in the Krebs cycle. Without those pathways, the people obviously suffocate. I don't know if it's like the movies. They always show people taking a cyanide pill. I don't know. I don't want to know. But even if you take a smaller amount, again, if it's enough to poison these reactions, we're in trouble. And so what they started after that is, you probably may not realize, but they started all this packaging. And so we've got foil on the inside, another containing a cover on the outside. And that's why I think we're all told, anytime there's a break in any of these packages, leave it alone. Because there was a time when the packages were sold in a very simple way, where anybody, in a sense, could pop a top open and do something, and that's exactly what happened. And then I think it was about 7 or 8 people are dead because of that. So, but what's the only interesting point I wanted to make, 2 things, I guess, is that, and plus the idea that you can't poison. We absolutely have to have this function. We can't thrive on this glycolytic pathway. Okay? Another interesting aspect of this, I mentioned it earlier, that each of us has maternal mitochondrial DNA. The sperm and the egg will unite, and there'll be what we call fertilization, but the middle piece, which contains the mitochondria in the sperm, is locked out, if you will. Only the head of the sperm with the DNA enters the egg and fertilizes it, and the end result, therefore, is what? That middle piece with the mitochondria from the male never gets there, and so it's all maternal mitochondrial DNA. It can be used for identification. It has some value there. Also, I don't know exactly how they use it, but if needed, they have, of course, DNA that we all hear about all the time, our DNA that we find in the nucleus, and then sometimes they can even use the maternal mitochondrial DNA for identification of in some cases where they need more information. Lysosomes. These are very interesting structures. They were discovered, as you can see, in 1949 by Dr. Christian de Duve. There was someone else who won a Nobel Prize along with him later. I forget whether it was in the early 50s, but he won a Nobel Prize for his work with these structures. I think part of it might have been, although this might have been later or it might have been at that particular point in time, but there's the lysosomes. They do a number of things, but they were at least implicated in muscular dystrophy. They didn't, a long time ago, they didn't really know that lysosomes cause muscular dystrophy. What was going on? We now know that muscular dystrophy, what I call the classic one, the deadliest of the types of muscular dystrophies, Duchenne muscular dystrophy, which affects males, sex-linked, and these males will die very early, 5 years of age, very early. Could be 5, could be earlier, could be a little bit later. There's no cure for it yet. And as I said, he and actually some of his students gained, I don't know the word fame, but maybe there's a little, at least fame in terms of the biology and physiological research, because he was studying these very structures that seem to be implicated in this disease. There's no doubt that with a person that has Duchenne muscular dystrophy, that the number of lysosomes will increase. Lysosomes contain over, as you can see, over 40 hydrolytic enzymes. Notice that word, hydrolytic. What's the implication with that word of hydro? Or how about both words, hydrolytic?
Something about water.
Okay, water, we got water, half of it. Lysis, breaking, right? Lytic, or breaking. We talked about dehydration synthesis. How about we add water back in? Chemically, of course. And that will split those bonds. Let's just say, take a peptide bond, for example. It was formed by dehydration synthesis. If we can add water chemically, we can split it. These enzymes will split bonds like that. And there's over 40 different enzymes. Now you can read somewhere that someone will say, I used to say 30, but then I found I saw it 40, whatever the number is. It's a lot of enzymes. We'll just go with roughly, say, 40 of these enzymes. Okay? One missing enzyme can be a deadly disease. And so there are quite a number, as you can see, mentioned on the next page, but there are quite a number of these diseases. The enzymes break down, ultimately, the lysosomes with their enzymes will break down materials that have been brought into the cell. These vesicles, remember we talked about, endocytosis. You bring things into the cell. You don't want it to just break open. So what will happen is the lysosomes will attach themselves to these vesicles and release their enzymes in a controlled manner and slowly, because of those hydrolytic enzymes, digest that structure. So let's just say we have a little vesicle. The lysosomes can attach, release enzymes into that vesicle, and if we had time-lapse photography, the vesicle would start to shrink inside. It doesn't pop open. That's not good. We don't want that material that's been brought into the cell just distributed any old place. So it's done in a controlled manner. Interestingly enough, I just mentioned in muscular dystrophy, there's an increase in lysosomes, which means they're breaking. There's a protein, I already mentioned it to you once before, dystrophin, which reinforces part of the cytoskeleton, and that's the protein that's missing in Duchenne muscular dystrophy. That, in turn, triggers these, in some way, these lysosomes to start splitting, breaking, and destroying the cell. That's called autolysis, self-destruction. So that's obviously not good when that's happening in the case of Duchenne muscular dystrophy. Following a pregnancy, let's say a woman has had her child, and after she's had her child, in case the guys didn't know, that uterus doesn't just snap back into place instantaneously, but rather it needs to be physically, something has to happen to it because it's there, it's palpable, if you will. It's enlarged tremendously, obviously, in order to carry the fetus to term. The lysosomes can take care of that. They can take care of excess tissue in the body in a controlled manner, so that uterus will then slowly, with time, shrink down to a more normal size, not perfectly normal, but much more normal than you would anticipate based on what you see at birth. And so the lysosomes can do that. You're going to get older. It's going to happen. I know all about it. It's going to happen. And whether we like it or not, we're not going to be as strong, our muscles, even if we decide we're going to exercise all the time and do that, we can't keep up let's say the strength levels, the intensity levels that we could when we were younger. Things are changing. We all accept that, and we know that's going to happen, but there are agents that take care of that. For example, no matter what you do, you're going to lose muscle fibers and muscle protein, but you're going to lose muscle fibers. You can take all the testosterone you want. Yes, that will do something. There's no two ways about that, but you're still going to lose muscle fibers. What's happening? The lysosome will self-destruct to some degree, and so slowly with aging, certain tissues are going to be broken down. Some of that very definitely involves these lysosomes. So they're perfectly normal structures. They've also been called, the person Dr. Deduve, who actually found them, didn't like this, but they've been called suicide bags. Because yes, when they break, the cell can be destroyed, but that could be a normal part of life, depending on what event is occurring. Obviously, in muscular dystrophy, we don't consider that normal, but in aging, returning the uterus to a normal size following a pregnancy, those are things that are acceptable and are going to happen. But obviously, in disease situations, we don't want to see that happen, but it does, because you have to have all 40 of those hydrolytic enzymes. In this diagram, you can see that the lysosomes are pinching off from the Golgi apparatus, or Golgi body. So this Golgi body needs to be, shall we say, really investigated a lot more. I don't know exactly how much has been investigated. At the end of this semester rolls around, I think I mentioned this, and you need to write a paper. Some of you will, you don't have to, but you'll say, I want a few more points. Any of these organelles would be an excellent choice. I'm not saying you can find, I think you should be able to find enough on any organelle, just about any organelle, actually. But not any organ. The appendix will take you down not a very good road. You'll be stuck trying to find information there. But if we take any of these organelles, these little organs, you'll be able to find a lot of information on this. So that Golgi apparatus is quite amazing. It's a temporary storage area, but it also modifies, as you can see, the structure. It also does what? Packages the 40 enzymes in a nice, neat little package, and then sends the lysosomes off. I forget the number, but it's in the hundreds of lysosomes that are present in a typical cell. They're supposed to be there. There's another organelle that was discovered by Dr. Deduve, peroxisomes. So he likes those small structures. I know a little bit more about this. Deduve used a special technique. He used ultracentrifugation to concentrate these structures because they're very small. Remember, he found them or discovered it in 1949. I know it's a long time ago, but still, it's a very small structure. You can't see it with a light microscope. You can't just go into a laboratory and say, Okay, you have an electron microscope, I'm gonna collect some lysosomes to use. How do you get them? He had a special technique to use what we describe as an ultracentrifuge, means it spins incredibly fast, kind of dangerous once it gets up and running. And he was able to set up gradients in that and get a fraction, a place, if you will, where the lysosomes, you might say, would congregate based on their density. And obviously in his work, he was working with small structures, and the peroxisomes, if you look at the size, and I bet the density too, is almost the same. So he was able to collect the lysosomes, obviously, report on them, win a Nobel Prize, and he was also able to go on and discover another one of these small structures in a cell that's very important. They contain not the hydrolytic but oxidative enzymes. It transfers hydrogen from various substances to oxygen, producing hydrogen peroxide, which in itself is toxic, so we need an enzyme, catalases, in order to convert the hydrogen peroxide to water. Zellweger syndrome is the most common peroxisomal disorder. How do we get that? One enzyme, wherever, I don't know the exact number, what all it takes is what? One enzyme, and you're going to get a genetic defect that can be quite significant. So Zellweger syndrome, you do not have to memorize it, but it's the most common peroxisomal disorder. Notice it shows itself at infancy, so the parents would know. It's estimated that is rare, 1 in 50,000 to 1 in 100,000. Results in death typically within the first year of life. There are other forms, so there are several different forms, and still death at an early age. The diseases result in the buildup of long-chain fatty acids. I don't know if this one or another one, but I remember there was Lorenzo's Oil, a movie based on a book of a family who had a child that had basically, I think, a peroxisomal disorder, and they developed a special oil, a fatty acid essentially, that was able to prolong that child's life. I don't know enough about it, whether it's exactly this, but I have a feeling that was a peroxisomal disorder. Whoa, what did I do? I went into that. We go back just a little bit. Centrosomes. And I really don't have a lot to say about them now. We're going to take a look at centrosomes again when we get to DNA, which is just around the corner. So I'm going to simply say they're involved in cellular reproduction. A centrosome consists of 2 centrioles. And the other thing I will say is that the fibrils and myofilaments of a cell are formed from these centrioles. And we're going to leave that there and move on to vaults, which were discovered in 1986. I don't know if they look like that, but I like the picture. I haven't found many books showing vaults. I was a little disappointed. I heard about them, read about them, started looking for them, and sure enough, you can definitely find a lot of information on these structures. So it's not like they don't exist, but I haven't seen some of the books mentioning vaults. But they're very interesting. Their size is significant and their shape is significant. This shape, either end, is basically the same shape as the pore in the nucleus. It's like a perfect fit, like a little spaceship in a way. So the belief is, perhaps, that it moves RNA from the nucleus to other parts of the cell, because we really haven't explained how the RNA moves out of the nucleus and just happens to find all of these different places in the cell to be located. And so the thought is that these vaults are involved in that process. Is it 100%? I have no idea right now, but